The chair for the afternoon session is Cooke, who's a professor in biomedical physiology and kinesiology. He's also an alumni of this meeting format, so he knows it well.
Thanks, Randy. And our first speaker is Nicholas Weilinger, who is research associate in the MacVicar Lab at the Center for Brain Health at UBC. He obtained his bachelor's at McGill and PhD from the University of Calgary, where his thesis centered on understanding neuronal cell death pathways during ischemic stroke. I will turn it over to you, Nick.
Thank you very much. Thank you for the introduction, and thank you very much to the organizers for giving me the opportunity to speak today. I will just quickly disclose that I have a position at a company, but this is completely separate from what I'm going to talk about today, which is understanding the ionic basis of swelling in neurons, and so how neurons can transform from what they normally look like here with beautiful arborized dendrites peppered with synapses to, in the case of cytotoxic edema, where you actually lose all of these synaptic features and elements, and they essentially collapse, and you get this sort of sausage link bead on a string phenotype, which is a hallmark of dysfunctional dendrites. And so cytotoxic edema is basically just cell swelling, so brain swelling. And so brain swelling is a serious medical issue, and it's a hallmark of traumas like stroke and TBI. And it's very serious because, of course, your brain is enclosed in a rigid skull. And so when it starts swelling, it has nowhere to go but down, and this can lead to death by herniating the brainstem. And so this is just an example here of a patient suffering from a hemorrhagic stroke that's leading to edema. And you can see here on his right hemisphere that you're sort of losing the typical demarcations that you see in the cortex. And it's actually swelling and sort of pushing against the midline and expanding into the contralateral side. And so our treatments for this are very morbid. It's the patient here underwent a hemicraniectomy to basically remove a window into their skull to allow the brain to expand outwards. And then the surgeon just puts a cap over top of the skull, which stays there for days, weeks, sometimes even months. This can persist, the swelling. And so it occurs as a consequence of energy perturbations and dysregulation in ionic gradients. And it's typically attributed to astrocytes. Astrocytes, as Brian mentioned, are the most abundant cells in the brain. And they have aquaporins, or water channels, that allow water to pass freely across their membranes. But neurons also swell. And so this is an example here of a hippocampal slice. Swelling is induced by applying veratridine, which opens sodium channels or keeps sodium channels open, and this can drive swelling. So that's a tissue level example. We can also look at the single nerve cell level. So these are neurons expressing GFP. Veratridine is applied. The nerve cells start swelling quite aggressively, and the swelling is so intense that it's actually very difficult to maintain focus. So neurons do swell, but how does this happen? And so it's actually been known for a very long time that the key to neuronal swelling, has been known for since the 80s, is influx of chloride ions. And so most of us as neuroscientists, when we think about chloride, we actually think about chloride as an electrolyte. That is, when chloride influx permeates through or conducts through chloride channels, we get a hyperpolarization or an inhibition. And this is all made possible by the presence of chloride transporters like KCC2, which uses the potassium gradient, the outwardly directed potassium gradient, to pump chloride out and keep chloride very high on the outside. But chloride is also a very important osmolyte. And the reason for that is, in the case of swelling, if you have, say, an excitotoxic event where you have persistent sodium loading, that sodium loading is often counteracted by, from an osmotic standpoint, by potassium efflux to hyperpolarize the membrane, leaving a sort of equimolar change. And so chloride is basically the odd ion out here, so that when it comes in and loads the cytosol, it makes it hypertonic relative to the extracellular space, and water can drive water can enter the cell that way. And so for a long time, it wasn't known how chloride gets into neurons. And in 2015, Ravi Rungta published a paper, who was a PhD student in Brian's lab at the time, identifying the importance of this chloride channel SLC26A11 to drive this process. And what was interesting about it is, unlike a lot of other excitotoxic cascades that lead to neuronal death, this did not require calcium entry. And an open question that stemmed from this work is, at least when I joined the lab, is this same process going on in the dendrites? Is SLC26A11 an important conduit for chloride entry in the neuronal dendrites? And I was interested in this because this sort of tied into my PhD work, where I studied blebbing, just to the extent that I was interested in using it as a surrogate measure of dendritic dysfunction. And so this dendritic blebbing is a reversible process. It happens during excitotoxic insults like ischemia, or you can trigger it directly with NMDA, like I'm showing here. And this prolonged blebbing leads to synaptic death also. So it's important when we think about models of stroke, or actually in the clinic of stroke, and when you have persistent swelling, that these persistently swollen neurons, obviously it can't be good for them, right? And so understanding the ionic basis of how this occurs was of interest. But there's a technical challenge to do this, and that is illustrated quite nicely in this video, where we have GFP expressed in the cytosol of these neurons. And you can see that as they swell, the signal is diluted, right? And so there's a technical hurdle to be overcome here, because if you try to do functional imaging of, say, calcium, or even using chloride sensors, you're going to get a false negative readout, because the dye is basically being diluted, and so the intensity of the signal is dropping. And so the reason, or the way that we circumvent this problem, is to use a technique that Brian mentioned in his talk, which is FLIM, or fluorescence lifetime imaging. And just to very quickly differentiate it from normal intensiometric imaging, where you are basically imaging the amount of photons that are released from a fluorophore that you excite with your laser, you instead measure this difference where an electron in the fluorophore is excited in this up state, and I call it the hang time. And so if you know exactly when the excitation occurred and exactly when the emission happened when you collect the photon, you can determine what the lifetime of that excitation process was. And the reason why this is important is because it's a quantitative measure. And so if we use a chloride sensitive dye to image chloride maps in the dendrite, for instance, you can calibrate this dye to different concentrations of chloride to give you reliable lifetime readouts depending on the chloride concentration. And these lifetime readouts will not change regardless of the concentration of the dye. And so if we have a dye dilution in the case of swelling, for instance, this will be unaffected. And so once we figure out what lifetime corresponds to what chloride concentration, we can just fit this with a mono-exponential decay curve. And then when we're doing the experiment, we say, oh, 3 nanoseconds is the lifetime, and so we can figure out that that's about 15 millimole chloride. So what does this actually look like when you do the experiment? So this is brain tissue that's bulk loaded with this MQAE dye, and so we can very nicely see that both neurons and astrocytes are loaded. And when we do these lifetime readouts, we actually get expected chloride concentration values when we do the FLIM calculation. And so in neurons, it's sitting below 10 millimole, and in astrocytes, it's about double that at rest. But what you'll notice from these recordings is, you can't really glean much in terms of dendrite morphology here, because pretty much everything is loaded. And so if we want to take a more specific or closer look at what's going on in the dendrites, what I tried to do is patch fill nerve cells with MQAE at 20 millimole, which is higher. This is more closer to what we're measuring in the astrocytes. And I mean, first thing, we can actually measure the chloride concentrations in the dendrites, which is great. But one thing that really jumped out at us was the fact that when you compare the measurements of your fixed chloride concentration in the pipette versus what you're actually measuring in the neuron, it's still drops back down to the physiological concentrations. And that's because neurons are notoriously good at maintaining their chloride gradients because, of course, this is what governs inhibitory tone in these cells. And so we can confirm that we can measure dynamic changes in chloride and dendrites. And so here's an example of a patch neuron. We can measure, we can record these MQAE chloride concentrations in the dendrites. We can increase chloride if we block the chloride extruder, KCC2, with furosemide. And so we get that expected chloride increase. We can increase it further still by depolarizing the membrane to open voltage-gated chloride channels. And both of these effects wash out very nicely. We can also do the opposite. So inhibit the chloride importer in KCC1 with bumetanide. And again, we see this expected effect, and we can, again, increase the chloride here by depolarizing the membrane. Okay, so that's just showing that the technique works. So what about actually measuring chloride changes in swollen dendrites? So how is this happening? And so this is now, this is a zoom in of a dendrite of a patched nerve cell. And here we're applying NMDA to trigger blebbing, using a trick to kind of limit the swelling to this single cell that I'm patching. And so what immediately should jump out at you is, so again, low chloride is the cooler colors, and in red is very high concentrations of chloride, upwards of 70 millimole and beyond, is that you see sort of the evolution of these formations of these, what we call chloride microdomains or hotspots, where the chloride concentrations seem to rise in a very sort of stereotyped way. And in the spots where we get the most chloride influx, that's where we actually get swelling, and these dendrites, these swollen dendrites, can actually lyse. And of course, we know chloride is involved. And so if we remove chloride from the perfusate, we of course don't get any chloride loading, and we also don't get swelling. And so this is just summarizing this here, just showing very nicely how you can see the evolution of these sort of chloride hotspots on the dendritic span here. So how much more chloride is there in a bleb versus in a non-bleb dendrite? And so if we compare two regions of interest, one dendrite or part of the dendrite that blebs versus an immediately adjacent part that doesn't, we can see that the chloride concentrations are almost double, reaching at peak on average upwards of 80 millimole. And we can do sort of a spatial analysis of what this looks like. And so this is before NMDA, during NMDA, and further along during NMDA after the full evolution of the bleb. And so what you can see is that there's a really steep cutoff at the edge of the bleb where chloride is very high within the varicosity. And just adjacent to that, it's almost 30 millimole lower. And this is over a span of, on average, a micron. And so there's clearly something's impacting chloride mobility along the tube, along the dendritic span here. That's basically keeping chloride in this specific location that's causing it to swell up like this. So I wanted to know what was driving this process. And so doing ion substitutions, again, you remove chloride, you don't get any chloride increases or swelling. You remove sodium, you get some chloride increase, but you don't get swelling. But what was most interesting to me was the fact when you dramatically reduce calcium in the extracellular perfusate, you get some chloride loading, but it's dramatically reduced, and the swelling is also dramatically reduced. When you inhibit the chloride transporters, I was kind of surprised because I saw the opposite effect of what I was expecting. And so again, inhibiting the chloride importer, which you'd think would block chloride influx in this case, it didn't do anything. On the other hand, inhibiting KCC2, the chloride exporter, actually blocked this very well. And so these data are just summarized here. So this is just an example of an analysis of how the extent of swelling in the dendrite. And so we get swelling in NMDA. It's reduced in low chloride, low sodium, low calcium. by inhibiting KCC2 with both furosemide and a more specific inhibitor VU02405551. And the steady state chloride is also reduced in the same way. But again, when we're trying to think about the mechanism and the relationship, it seems like there's an involvement in KCC2 and there's an involvement in calcium, which is different from this somatic mechanism. So what's going on here? And so you can do calcium FLIM imaging, much like you can do chloride FLIM imaging. And so this is with the calcium sensitive dye OGB1. And so if you just depolarize the cell, you get very nice calcium increases that, and this is depolarized for 40 minutes. And so you get sort of a steady state plateau that doesn't really change over 30 minutes and you don't get swelling. So depolarization alone is not sufficient to drive this process. But when you combine depolarization with NMDA, you see the same sort of calcium heterogeneities along the span of the dendrite like you would in chloride. So wherever you have the highest calcium concentrations is where you get the blebbing. And the threshold for this, or the concentration of calcium in these regions that bleb, is above half a micromole, sometimes upwards of 1 micromole and beyond. And so we started to think about what could be going on here. So there's a role for calcium, there's a role for KCC2, there's a role for NMDA. How could these be linked? And so one thing that jumped out was the calcium activated small conductance potassium channel, or SK channel, which you might have learned about in your first year neurophysiology class. And this is a calcium activated potassium channel that has interesting associations with the NMDA receptor. So it's located right alongside NMDA receptors in the spines. And so there's a physical association. There's also a functional association where they open right after NMDA conductances occur. And they actually opened right around that window of calcium concentration that we measured using the FLIM signal. So about 500 nanomole. And they're voltage independent. So just performing a sustained depolarization is not enough, or it will not open these channels. And so luckily, there's a highly specific peptide inhibitor called apamin that blocks SK channels. And so when we bath apply apamin in the same way in these experiments, we again see a dramatic reduction both in the chloride loading and in the swelling. And so that's just all characterized here. And so that leaves us with the mechanism of how this all works, where we stray from an equilibrium situation in NMDA stimulations, like what you would get in stroke, where you have overactivation of the NMDA receptor, sodium loading, calcium loading, and this calcium influx activates the SK channel, which leads to a high concentration of extracellular potassium. And so I told you that KCC2 relies on this potassium gradient to keep chloride levels high on the outside, but the transport can actually flip, of course. So if extracellular potassium levels rise, you can actually get reverse transport of KCC2. And when you get the combined loading of sodium and chloride, it becomes the dendrite becomes hypertonic and then swells. And so we think that this is kind of a cool paradigm for compartmented necrosis, which is of course relevant in neurotraumas, but perhaps also in neurodegenerative disorders where you see spine loss. And so I'm just going to very briefly go into how this relates to swelling, only because Brian teed it up. And so swelling, of course, can cause synaptic death, like I just showed, but it's also sufficient to cause, complete death of the neuron. And so coming back again to the Viratridine model, where we induce swelling at the slice level, we can analyze this very nicely, where we can monitor the swelling. It's reduced in low chloride. But what about the actual death? And so swelling itself is sufficient to cause cell death. And so we were very interested in testing the hypothesis that this large pore channel called pannexin-1 might be involved in this, because pannexin-1 opening is known to contribute to cell death in ischemia. And so this is a large channel that has these C tails that loop up to block it, and it can be opened either independent or dependent on caspases that cleave these tails. And it's permeable to all sorts of ions and large metabolites. And so when we do various cell death assays in this swelling model, we can see that blocking pannexin is protective in this viratridine-induced swelling by LDH release. We can also image, these are neurons loaded with propidium iodide to label cell death. And when we inhibit, again, pannexins with carbonoxolone, which is a widely used blocker, we can also block cell death in this model. And so pannexin is known to cause cell death, but it's also an ATP release channel. And so we heard a lot about microglia so far today, and I think there's more to come. But Brian very nicely showed that microglia are attracted to sources of ATP. And if pannexin is opening to cause cell death, maybe it's also releasing ATP to attract microglia to these dying neurons. And so that's exactly what we saw. And so this is a neuron that's patch loaded with a hypertonic internal, so it drives water entry or osmotic swelling. And you can see that the microglia are particularly interested in this cell. And that's because they are compared to control. So microglia are always surveying their environment, and so they're always contacting neurons, but the contact rate in swelling neurons is much higher. And so that's illustrated here, and they have the same amount of initial contacts. And so this is just a quantification of the actual contact rate. And this contact rate is reduced, again, when we inhibit pannexin. And so because pannexin is causing death, but is also an ATP release channel, we wanted to play around with this signaling axis in microglia. And so these are recordings from mice that were fed a PLX diet, which is a way that you can deplete microglia in the mouse brain. And so we get a very nice depletion. About 80% of microglia are gone, and that's what we see here. And when we repeat this experiment to look at PI-loaded neurons or dead neurons, we can see that the actual PI-positive cell density is higher, meaning there's more dead neurons in this assay, suggesting that microglia are in fact playing a protective role in reducing the number of dead neurons. And so we can assess this axis in another way. So instead of depleting all microglia here, we're inhibiting P2Y12. These are the purinergic receptors on microglia that are detecting the ATP. And so when we block P2Y12 receptors with this PSB compound, we both see an increase in cell death. So there's more neuronal death when we perturb this purinergic signaling. We also see fewer enwrapped PI-positive neurons in the PSB, which makes sense because the microglia are having a harder time detecting that these neurons are dead. And so where I'll leave it at here is the actual mechanism of all this, where when we have a swelling neuron and edema, we have normal microglial surveillance that's kicked up when they start releasing ATP and signal to microglia. And that's all because pannexin-1 is sort of playing a dichotomous role where it's both killing the cell, but it's also calling for help. And so it's kind of a double-edged sword, because if we inhibit pannexin, we might, we'll improve, we'll reduce cell death at the single cell level, but it also prevents or perturbs the mobilization of microglia processes to these dying neurons to exert what a lot of people are calling a soothing touch, where they can actually protect nerve cells. And so I'll end it there. I'd like to thank everyone who is involved in this work, Ravi, who did the initial work, and then Lee, Jeff, and Chris, who helped me with this project, and of course, Brian's supervision and my funding sources. Thank you.
Thanks, Nick. That was a really interesting talk. In terms of the application of this knowledge is what I'm really curious about, and maybe you could speak about. What would you see this as something that someone were to get a stroke, we could block this, or immediately after a TBI, we could potentially be targeting these pathways to prevent the swelling? Is that kind of, if we're thinking about the translation of this?
Yeah,
That
yeah, absolutely. Thanks for the question. So it's a good question. And the reason for that is, as I said, our treatments are quite morbid and they've been around for decades. And so in addition to hemicraniectomy, osmotherapy is often used as well, where they'll basically just inject a concentrated large sugar like a mannitol systemically. And the idea there is to drive water out of the brain and into the blood compartment. But what often happens in the case of stroke and TBI, like Brian illustrated, is during these traumas, you get permeabilization of the blood-brain barrier. And in those cases, mannitol can start leaching into the brain compartment. And then that can actually make things even worse. And so I really view this as more of a starting point, because there really isn't any way to tackle this problem other than these two treatments that, they can work, but neither addresses the actual mechanism of swelling itself. They're ways of accommodating the swelling. And so it does present a problem, though, of course, because NMDA receptors, this has been tried, like inhibiting NMDA receptors in clinical trials for stroke. This doesn't work. KCC2 is ubiquitous in neurons. This is a really important transporter under physiological conditions. And so inhibiting this, this can lead to epileptiform activity and things that you don't want. Maybe the SK channel could be a target, but I guess I'll just say that it would be nice to come up with some therapeutic intervention that could disrupt this pathway, but it would be pretty difficult, and it would perturb a lot of important physiological function of these three players. And so maybe a more nuanced approach where, all three of these are modulated by kinases, and, it could be that you could alter their function in a more nuanced way that's not on or off, that maybe would be a little safer. But yeah, unfortunately, I'm hopeful that, studying these types of pathways would lead to new therapeutics, but I don't know if we're going to do much better than hemicraniectomy and osmotherapy for a little while until we understand this a little better.
They could probably hear me from there.
I can.
Yeah, so many thoughts in my mind, but I kind of want to go off chart, talk about the TBI stuff later. But one of the big issues that we have for doing really good, histology and histochemistry is to combat that shrinkage in tissue that comes due to water loss, et cetera. People have been playing with systems doing pressurized perfusions, doing sucrose, preload, postload, personnel, et cetera. Do you think there may be an application here for
Yeah, that's a good point. Actually, I got to say, I don't do a lot of histology myself, and so I'm aware of these problems, but it's not something that I've thought a lot about. But I could see potentially inhibiting some of these transporters, that it might impact water movement a little bit. The thing is, especially in nerve cells, it's still not really known how water gets in. So we always kind of hand wave over this, because a lot of these transporters, they do carry water. They kind of piggyback with these other ions, but so this would influence, osmotic water entry, but we still don't know how the water gets in or out. And so probably the best way would be, once we figure that out, would be to block that process.
Yeah.
But unfortunately, I don't think I can really help you beyond that. But it might be something worth trying one day.
Yeah.
That was a really wonderful talk. Thank you. One of the questions I had was about some of the regional differences where you've got areas of Fleming, adjacent areas without. Obviously, the movement of these ions can be very fast across. There should be a lot of diffusion across. What's creating these microdomains, and what separates out areas where you're seeing splitting versus those that don't? Because that itself might be a protective mechanism, sort of walling off those areas.
Yeah, definitely. So the short answer is we don't know, but there's hints coming from the literature, and even that though is a bit contentious. So one thing that happens, and actually it's illustrated here, is when you get this sort of collapse of the synapses and it all sort of becomes this big swollen varicosity, the actin filaments that are usually in the spines, they start breaking down and clump together. And so there was a nice Nature Communications paper about this recently. And so it's possible that a combination of maybe membrane pinching and or clumping of these actin fragments, which would be charged, might impede diffusion along the cable. And I mean, from a resilient standpoint, it's actually debated whether or not this blebbing might actually be protective in itself as a way to, sort of compartmentalize the excitotoxicity, because actually I do have a slide about this too. Synaptic death. So this is just an example from Tim Murphy's lab, which I reference a lot, because this is an example of I think these are barrel cortex neurons expressing channel rhodopsin. And basically what the point of showing this is that the neurons are still alive, but they're stripped of their synaptic connections during ischemia. And so the neuron, after reperfusion from ischemia, you can still drive action potential firing by stimulating these neurons with channel rhodopsin. But if you try to stimulate them by evoking some forepaugh stimulation that would rely on synaptic connections, you can't do that. And so the neurons are alive, they've just lost their synapses. And so from a resilient standpoint, it's an interesting idea that this might be a way of, it's like kind of sacrificing the part of the dendrite that's dying to keep the rest of the nerve cell alive. And so, yeah, that's, again, not really known how this compartmentalization happens. I initially thought maybe it's different NMDA receptor subtypes, or maybe the localization of NMDA with KCC2, but they do seem to be pretty much everywhere. And so I'm not sure that it's that. It might be something that has to do with impeding the mobilization of these ions, which is interesting because even myself, like, we usually think that, okay, they're freely flowing ions. Of course, they could just diffuse down the dendrite, but it doesn't seem to be the case, at least in this pathological setting.
Like a storm drain, is that way?
Yeah, kind of like a storm drain that's getting blocked. Yeah. But this also happens at rest. So here's an example in calcium. Well, this is like one of the earliest examples. And so this is an experiment by Baker and Ransom, where, phenomena like this have been seen before, where you can puff on glutamate and GABA. This is a cultured neuron in different parts of the dendrite, and you get completely different GABA responses or polarity of GABA responses depending on where you puff it. And so even at rest, it's possible to have different chloride microdomains in a nerve cell. And I think I have another slide on this too. This can happen at rest in calcium as well. So this is an example from Dmitry Ruzikov's lab, where you can have regions under physiological conditions of high calcium concentrations that persist over several minutes. So you can have compartmentalization in spines, or even just a spine and a couple neighboring dendritic regions. And it's not known how this happens either.
I guess I'll ask another question then. In terms of the, so these are obviously excitatory neurons you're showing, right?
Yeah.
So as you mentioned, there's a very robust inhibitory intraneural milieu in many cases that's supposed to be protective in these particular cases. I don't know too much about your model system, but I thought if you want to take a second, maybe comment on how you think this reflects the physiologic state in the presence of inhibitory tone being present.
So do you mean under more physiological conditions, or in a more extreme case like a swelling case?
So normally, a lot of these synapses would be surrounded by inhibitory interneurons. Obviously, the
Yes.
calcium buffering is very different in those intraneuronal systems. But how do you think that might impact some of the formation of these microdomains, or the fact that you're sort of, have you stripped that out? Have you stripped out inhibitory? Are there any inhibitory neurons in your culture system?
So actually, all these experiments, I think I failed to mention this, that's all in acute slices.
Okay.
So there are inhibitory interneurons there.
You have like bicubulin in there or something like that to take out the tone? Or?
So I actually don't, but I did experiments to block GABA receptors, and surprisingly, it had no effect. And so what I would say to that, I'm just going to bring it back here. What I would say to that is it does get me thinking a lot about neurodegeneration, where you might have perturbations in chloride transport. There's evidence that A beta can impact KCC2, and so it's perfectly reasonable to think that inhibitory tone in a dysfunctional neuron can switch if KCC2 function is perturbed, or maybe if it doesn't even need inhibitory tone in that spot. If there's enough excitatory tone, which can indirectly impact chloride gradients, you might get a similar situation where you have elevations of extracellular potassium that could flip KCC2 and cause chloride entry on its own. And so that is something that we've been thinking about and that we've been wanting to study, actually, is how all of these things might be interrelated. Inhibitory tone changes in KCC2, and how KCC2 might be impacted in neurodegenerative models.
Right, so following up on the actin filament disruption, so does it mean there's any interruption in terms of the anterograde and retrograde, like cargo trafficking at the swollen site?
I don't know, but I would assume so. So, I mean, it could be charge dependent, but the fact that we see both calcium and chloride being sequestered, like here, sorry, like this, right? Like, I don't know. I feel like this is very striking, the fact that calcium cannot diffuse. And so I think if calcium can diffuse and chloride can't diffuse, there's no way you're going to get any sort of protein shuttling down to dendrite. But that's, he's speculating. It would be interesting to test.
All right. Thanks. Let's thanks Nick one more time.
Thanks, everybody.